my apologies to everyone for the technical problems, but we'll just go ahead. And I understand that some of you may have to leave because we'll go over schedule now, but let me begin. So the charge today is for us to talk about tick diseases, or deer ticks and associated diseases. And let's get on with it. To start, I just want to emphasize that there are some excellent resources out there, including tickreport.com, our own webpage, but also the Tick Encounter Resource Center, which is a very thorough resource for information on deer ticks and other tick species, and some information as well about the associated diseases. Later in the presentation, I'll give you some links to the CDC, which is another good source for information more about the pathogens themselves. As I tell students often, my job as I see it is to weave together a story with facts, and the facts are usually available in a disparate number of places, and so you can find them on web pages like this. And I bring this up because a couple of you since the last webinar have emailed me and said, what was meant by that that figure or this point? And I'm happy to respond to those questions. I also in this place have tried to provide linkages to the actual data where you can go back and follow these up. But Tick Encounter is an excellent page to go to. So to begin, let me talk about ticks in general. So it turns out there are not just a few tick species, there are actually hundreds of tick species, and they can be split up into major groupings that are sort of indicated by this this evolutionary tree diagram here. And I'll just show at the bottom the that there are these ticks called soft ticks, which are not generally of concern to us because usually these are ticks that feed on their host, either in the host or in the nest or the burrows of the animals they're associated with. And as such, they usually don't bring, even if they contain pathogens, they don't bring them into our realm. The two groups at the top are hard ticks, metastriate and prostriate. I'll start with the metastriate, which includes, and I've given Latin names here, my apologies for that, but these are the things like the brown dog tick, the common dog tick, the lone star tick, the cattle tick. Somewhat less common here in the Northeast than other parts of the world or the country, but they are nonetheless hard ticks. They have a number of similar behaviors to deer ticks. In future webinars, we'll talk about some of those other species that are associated with disease transmission. The ones we're most concerned about today are the Ixodes ticks, which are members of this group called the Prostriata. And I want to emphasize here that there are a number of species of Ixodes in addition to the deer tick, which is canonically known as Ixodes scapularis. And some of those may be associated with transmission of some of the same pathogens, but as we'll see, because of ecological differences and maybe some biological differences, other biochemical differences, they may or may not transmit things like Lyme disease. So ticks are diverse, and we're just going to focus on one of them, one of those species, in particular the one that's represented here on the leftmost panel, which is the deer tick Ixodes scapularis. Two other tick species I include here because, like deer ticks, dog ticks and lone star ticks are of interest to us because they are human-biting ticks. So among those several hundred species that were represented in the previous diagram, a very small number of them actually have much choice for us as source of blood meals. And so these three species like us as a human host, and deer ticks in particular. So just a few things to navigate on this this figure here. There are three panels, obviously the deer tick, the dog tick, and the lone star tick. And what I'd like to draw your attention to is that there's 4 different specimens representing each of those tick species. And so what those are, going from left to right, bottom, and then up, are the larval tick, the nymphal tick, and then the adult tick. Jeffrey, are you still on there hearing this? There's someone with a microphone on in the back, so I was just trying to figure out. Is that you, Jeffrey?
I muted myself, sorry. I was.
That's okay. My question for you is, can people see this cursor when I use this here? Does that show up on your screen, the cursor that I'm showing on the slide? I've lost you again.
I can see it.
Thank you. So going from left to right, here's the larval tick, the nymphal tick, and then the two adult ticks at the top, the two representatives of adults. And it's important to understand that if you look across the different species, that all species have very small versions in terms of the larval tick. And so when it's said that deer ticks are the very tiny ticks and dog ticks are the larger ticks, it may be that people are making a comparison between this, a larval deer tick, and this, an adult dog tick, which clearly this is larger. But when one makes a comparison between the larval stages, larva to larva or nymph to nymph, you can see there's more, the sizes are more similar. My apologies for that again. And so there are three life stages: larval, nymphal, and adult. Another point to point out here is that as juveniles, which includes the larva and the nymph, there is no sexual dimorphism. In other words, the ticks, males and females, have not yet distinguished one from the other. Whereas as adults, they become distinguished as distinctly male, as shown on the left, or female, as shown on the right. And at each of these life cycles, life stages, the tick has exactly one job, which is to find a host, to feed on that host to repletion, which can take a day or two, or even up to a week and a half, and then to utilize the blood meal, to utilize that source of its nutrition to molt to the next stage. So the difference between these two species, or between these two stages, is the result of a complete metamorphosis. So they basically lose their entire exoskeleton and gain a new exoskeleton that builds around the midgut of the tick, and they use the energy from the blood meal to do that molting process. And I emphasize that to just reinforce the notion that this little tick right here will feed exactly once in its larval lifetime, and this little nymphal tick will feed exactly once in its nymphal lifetime, and the female will feed exactly once in her lifetime, and the male will never feed at all because males don't feed. That's important for disease transmission, or understanding of disease transmission, because what that means in terms of public health is if this tick started to feed on your dog and it fell off and got on your couch, in all likelihood, it may not have the capacity to initiate that feeding process again. So if she didn't complete her blood meal on the first host, that's probably the end of her. More importantly, she only really gets one opportunity to become infected, because for most of the pathogens we're talking about, they are born without infection. And most, in particular for Lyme disease, these larval ticks have no Lyme disease because they don't acquire it from their mother. They have to acquire it during the course of their lifetime, as we'll see in a moment. One more point to make about the images of the ticks here. You can see that there's different characteristics, coloration, shape, etc. And you can see that the deer tick and some closer related species that don't feed on people very often have very distinct shape in terms of the mouth parts are more elongate, but also in terms of coloration. They have this, particularly in the adult female, there's a clear orange area, reddish, brownish orange at the back margin of the tick, which distinguishes it quite a bit from the Lone Star tick and the dog tick, which we say are more ornamented. At the bottom of these images of ticks pictures, you see there are diseases associated with the different tick species. And these are pretty fast and hard. That is to say that Lyme disease really is not known from these other two species of tick. So too, ehrlichiosis, which is a pathogen associated with an Ehrlichia pathogen transmitted by Lone Star ticks, it's not known in dog ticks or deer ticks. So knowing the species of tick is fundamentally important to knowing the risk of pathogen exposure. The other point to take from these lists is that we're not talking about single pathogens, that ticks may be infected with one or more pathogens. In fact, we've seen ticks infected with up to 4 or 5 different pathogens in a single tick species, or a tick specimen rather. And so that's an important point for us to remember as well. And last thing I'll say about this slide is just that you should know that while all of these diseases below are associated with pathogens that can be found in these tick species, not every tick specimen has any or all of these things. That many ticks, in fact, as many as 50% of the ticks are not infected with Lyme disease. Perhaps as much as 98% of the ticks are not infected with Borrelia miyamotoi. So the infection rates vary for different pathogens, but the very simple message is that not every tick, regardless if it's the right species, is infected with every pathogen. The CDC and the departments of public health, in particular Massachusetts Department of Public Health, if one were to ask them the question of where risk of disease is greatest, they would tell you that, logically, that risk of disease is great where the tick associated with transmission of that disease is predominant. And so, for example, Lyme disease that's transmitted by black-legged ticks might be found in any of these areas where the black-legged ticks are found. Likewise, diseases such as tularemia that are associated with American dog ticks may be found in the range where the American dog tick is found, or pathogens associated with Lone Star ticks. And we'll talk about those other 2 species in the future. So a word about just behaviors. I may have showed this animation before, but it's so good to emphasize the behaviors that ticks exhibit. Ticks don't fly, they don't jump, they don't fall from trees. They crawl very short distances and seek a spot where they can quest. And we'll talk more about the different questing behaviors of different ticks in a moment. But she's crawled out this tick, which is a female deer tick, and she's waiting for a host to come by. And when she finds the host, she latches on with her legs, and then she crawls up to a spot where the skin may be thinner, or she may be protected. It might be a slightly warmer area of the body. And then she plunges her mouth parts into the host and glues herself into place and starts a very complex feeding process that involves spitting and spitting salivary juices, quite literally, into the site of the wound, but also sucking blood up. And it's a very delayed and very slow and regimented process that goes on. It's a physiological process. It's not simply like the tick, like when you go and give blood at the phlebotomist and the phlebotomist sticks a needle in and sucks blood out. It's much more complicated. There's a two-way process going back and forth, and you'll see in a moment why that's fundamentally important to our understanding of how pathogen transmission takes place in these ticks. Here's another diagram sort of showing that process of the tick feeding. She uses her mouth parts, which are called the hypostome, those hollow mouth parts, to create a feeding lesion. And then there's a two-way flow, a spitting and a slurping, that is the technical term for it, of fluids in and out of the feeding lesion. And what's being shown here is the midgut at the top, but as you'll see in a moment, that there's actually salivary glands, which are separate organs inside the tick, and the two are not connected directly. And when she's done feeding, she will detach, and if she's a juvenile tick, she'll fall off and use that blood meal to molt to the next life stage. If she's an adult female tick, she will never molt. She'll use the blood meal that she's acquired, if she's been mated with a male, to produce a batch of 3,000 eggs, 2,000 to 3,000 eggs, that will then give rise to larvae in the next generation. So different species of ticks have different life histories. And one of the ways that we distinguish the different tick species is by the number of hosts that they're associated with. So some ticks, for example, Boaphyllus micropus, not a deer tick, are associated with a single host. And the upshot is that that tick will feed on, in this case, a cow or cattle, all three of its life stages, as a larva, as a nymph, and as an adult. And the only time that it really spends off the host is the time when the female has laid the eggs and the eggs fall off, and she falls off, she oviposits, and then lays her eggs and dies, and then the hatched larvae will then get back on a host and then spend all of their lifetime until they lay eggs on that single host. But a single host species, that's the one host tick. Logically, the two host tick is slightly more complicated, where part of the life cycle might be spent on one host, but then they fall off and they spend another part of their life cycle on a different host. That's a two host system. Here's the example of Dermacentor andersoni. Deer ticks also follow this family. What distinguishes them from other ticks is they are three host ticks. And that's important to the disease transmission cycle as well, because they feed not just on one species, but they feed on at least two, in some cases, three different species. And so in the case of the deer ticks, they feed on usually small mammals or rodents in one life stage. They fall off. They may get on the same mammal species at another time, but a different animal, not the same specimen. And then as the third stage, as adults, they actually feed almost exclusively on deer. And the reason that's important to us is that each of those bites at the apple, if you will, is an opportunity for that pathogen for that tick to acquire pathogens from different hosts. And so what makes three host ticks particularly amenable to disease transmission is the fact that they're sampling different hosts and acquiring different pathogens, or have opportunity, I should say, to acquire different pathogens. And this was one of the problems, or one of the reasons that deer ticks pose such an important problem for humans. So I have a little video here to show, this is an actual video showing the ecdysis process. And I use this because I really want to emphasize that blood meal is a complete process, and they then utilize that for the sole purpose of molting into the next stage. So shown here are actually two larval ticks, which are blood fed. There's one with her belly side up, his or her, and here's one with the back side up, sort of looking down at the dorsal side. And as we start this video, you'll see the engorged larval ticks start to ecdyse, or to emerge. So this is a 10 times speed video, so it's sped up so we don't have to sit here. But you see that she actually emerges. The entire body, the entire tegument of this tick, with the exception of the midgut, is newly created. And so she or he has carried over just the gut contents from that first life stage to the next life stage. And that's fundamentally important to the disease transmission cycle as well, as you'll see in some future diagrams. Because of the 2-year life cycle of deer ticks here in the Northeast, where the eggs are laid in the spring, they emerge in the summer, usually late July through August into September, as larvae. Those larvae then will go and look for usually a small mammal, although occasionally on a bird. They'll take a single blood meal, and then they'll use that blood meal to ecdyse, just as we showed in the last slide. And sometime in nymphs, they will emerge as nymphs in the spring of the following year. So already almost 9 months have passed in the life cycle of this tick, and all she's ever done, or he's ever done, is taken a single blood meal. These nymphal ticks then go and quest their own blood meal, and they will look for small mammals, or in some cases larger mammals, or in some cases us. And if that larval tick fed on a mouse that was infected with a pathogen, a bacterial pathogen or another germ, that tick can then carry it on as a nymphal stage and then give it to the next host they feed on. And in fact, this is the reason that the transmission cycle is maintained in nature, is that larval ticks feed on infected mice, and then as nymphs they infect new mice so that the disease transmission cycle is maintained, because mice from this cycle will then give rise to, or feed later that summer, to larval ticks that are born uninfected. And then they carry that infection through to adults. And then as adults, because they are three host ticks, they feed almost exclusively on large mammals and predominantly on white-tailed deer throughout the range in the Northeast and upper Midwest. And then they'll lay the eggs and the cycle will start over again. And so the diagram is shown this way because it's very important to understand that although in this lifespan of a single tick, the larval stage clearly comes before the nymphal stage, just as the adolescent stage comes before the teenage stage. In terms of an annual cycle, spring to summer, the nymphal stages of a particular species are actually feeding prior to the larval stages of the next cohort. And this is the necessary phase for the transmission of the pathogen within these small mammal species, as represented by this gray box. It's because of that 2-year life cycle and the fact that the nymphs precede the larvae in a given year that we have Lyme disease cycle. Were we to break the 2-year transmission cycle and reduce it to a single year, it would be a very different scenario, because larval ticks are born without infection. They don't have Lyme disease. And so they would have to acquire it from a group of hosts that generally aren't there. This is an image from a paper a couple of years ago, and it emphasizes the increase in the distribution of Ixodes scapularis, which is the deer tick, the black-legged tick here in the eastern United States, and the western black-legged tick, Ixodes pacificus. And what's being shown here is in the red, these are confirmed, and then the blue are suspected distributions. And you can see that the red is increased in terms of this period from 1907 to 1996 versus 1907 to 1915, and 2015. And the argument being here that in the 19 years between this and this, you've seen this huge increase in the areas that are now established ranges for Ixodes scapularis. Interestingly enough, so if you can keep this image in your head and imagine and see the different states and the geography of the continental United States, you see the red distributed here in the northeastern United States. There's upper Midwest. There's a little bit of a lag here in between, and then you see down here in the southeast and to a certain south central distribution of ticks. The distribution of Lyme disease doesn't closely match that. So here you have lots and lots of Lyme disease. Every one of those blue dots indicates a single case of confirmed case of Lyme disease. You have a predominance in the northeastern United States, a focus up here in the upper Midwest, and then throughout the rest of the United States, there are some spots where there's virtually no Lyme disease, but even down here in Florida, where, as you recall, the Ixodes scapularis ticks were, just to go back, quite abundant, we don't see a lot of corresponding Lyme disease cases. This is important to know. Let me sort of tell this story a little bit going back. So historically, and by historically I mean prior to the 1970s, the distribution of Ixodes scapularis was as follows. It was basically restricted to the southeastern United States. And many of you will know the story of Alan Steer and the group of concerned mothers in Old Lyme, Connecticut, that reported the first cases, the first North American cases of Lyme disease, or the first epidemic, as it were. But we may not be as familiar with the fact that on or about the same time, Professor Andrew Spielman at the Harvard School of Public Health was investigating an outbreak of a tick-borne illness on Nantucket Island. It turned out to be babesiosis, which is still one of the diseases that's commonly associated with deer ticks. And Spielman made a note that this tick looked very much like Ixodes scapularis, the ticks from the southeastern United States. It was clearly disjointed. It was not in a contiguous distribution with the southeastern United States, and yet it looked very much like Ixodes scapularis, and after careful examination under an electron microscopy, he concluded that in fact it represented a different species, a species that hadn't been identified at the time. And so he identified that as Ixodes damani. By 1989, the distribution of that northern species, Ixodes damani, was as follows. Here you see the Ixodes pacificus ticks in the western United States. The distribution of Ixodes scapularis was in the southern United States. And then this new species that Spielman had described in 79, by 89, there were all these little pockets that started to show up. And it's unlikely that all of these were attributable to expansion of the range that quickly. It's probably that there were several foci. For example, this focus in Wisconsin, which was outside the town, initially found outside the town of Spooner, Wisconsin, was probably another refugial population, another population that had been left there after the last glaciation, recession of the last glaciers, and that then they expanded from these different foci. But the point is that there was a new species, and it had this non-contiguous distribution with the ticks from the south, and it was called as something separate, Ixodes damani. Later, largely for non-biological reasons, I'll say, the two species were unified under a single name, Ixodes scapularis. So at one point, the name of Ixodes damani was canceled, and now it's not widely used. My friend and colleague Sam Telford still uses the term Ixodes damani. It's still recognized as an important subspecies. But more importantly, with the loss of that name has been a loss of the recognition that in fact there's biological differences between these northern species and southern species. And it's that choice to sort of overlook those biological differences that creates this lack of congruence between the distribution of Ixodes scapularis in the eastern United States and the associated distribution of Lyme disease. Most Lyme disease is centered in the places that we've known ticks to be since 1989, and it is expanding from these into the rest of the northeastern United States, but it's clearly not associated with the species that was previously known as Ixodes scapularis. One of the fundamental, sorry for the title of this slide is below the image, but one of the observation that was made by a group from Wisconsin a few years ago in a paper in 2015 was a behavioral difference. It was found that when they took ticks from the south and ticks from the north, and then they put them in these very interesting outdoor arenas where they allowed the ticks to do whatever they would, they observed questing behaviors. That's the crawling out onto the edge of a branch, or an artificial branch in this case. They found a difference in the questing behaviors of the northern and southern ticks. So here you have ticks from North Carolina, South Carolina, and Wisconsin exercised on these different arenas in either the South, Florida, Tennessee, or the North, Rhode Island or Wisconsin. And what you see is that regardless of whether they're in the South or in the North, the southern ticks, the ticks that originated from the south, have a greater questing behavior. And the slide at the bottom is meant to show that this wasn't attributable to just the hardiness of the ticks. In fact, the survivorship was roughly the same across all the species. And so these authors had argued that one of the reasons we don't see Lyme disease in the south is that the ticks don't quest the same way in the south that they do in the north. And it's that questing behavior, as we showed in the video, that leads directly to the transmission of Lyme disease, because if ticks don't quest and bite humans, then they cannot transmit the pathogen. So more generally, going back to figuring out what makes ticks such great vectors, as it were, or transmitters of pathogens, we have to look a little bit at the tick physiology. And I'm sorry, this is quite a busy slide, but it shows some important things. Shown here is the tick. So here's its legs, if you can make those out. And then there's an outline, a cutaway of the tick's body. And inside you see this brown area labeled MG, that's the midgut. And then you see these little balls here. These are the salivary glands. And then the lighter colored spheres or balls here are actually the ovaries. And below it is the red area meant to represent that feeding lesion. What's happening here is they're showing that as ticks feed, they pick up pathogens represented by these little blue blobs here and the arrows indicating the direction. Some pathogens, as they move from the feeding lesion, so everything below this brown line is the human host or the mammalian host, perhaps a mouse, that as these little blue blurbs move through the tick's mouth parts and into her midgut, they end up in the midgut, inside what is ostensibly the some mixture of the stomach and the large intestine of the tick. They don't have stomachs proper. And from there, they may do one of two things. The pathogens may stay in the midgut, which is a bag sealed within that body, or they may make the migration during the course of a single feeding out of that midgut and into the salivary glands. And this is an important biological process going on here, because again, ticks aren't just bags or syringes that are sucking blood and or spitting saliva. There's physiological changes that go on inside the tick. And just as your organs, your heart is separate from your liver is separate from your kidneys, so too the midgut is a very separate organ from the salivary glands or the ovaries of a tick. And so for a pathogen, a bacterial pathogen, for example, to come out of the inside of the midgut and go into the inside of the salivary glands or the ovaries, it has to traverse a pretty toxic environment, which is the tick's blood or the hemolymph of the tick. And it's a toxic environment because the tick has defense systems just like we do. Just as our blood is full of defenses, although they don't make antibodies and we make antibodies, the tick is armed with a number of antimicrobial peptides and other things that it uses to protect itself from infection. And so the bacteria have to be very cautious as they make their way out of that midgut, where they're basically safe with the rest of the blood meal, through the very hostile hemolymph and back into the salivary glands. And so to do so, they have to change expression of different genes. They have to change different coat proteins. They are not simply the bacteria, the same bacteria that was acquired by the tick. And Lyme disease is a perfect example of that. The spirochete actually changes, the bacterial spirochete associated with Lyme disease actually changes the protein coat as it moves from the mammal host into the tick host. And then it does it again as it moves from the tick host and back into the mammal host. The other aspects of this diagram, just to finish it out, are this process of moving from the midgut to the salivary glands, as shown in the bottom figure. It can take place during the blood feeding, or it may take place after the blood feeding is complete, after the tick has taken its blood meal. And then the pathogens can move, for example, to the ovaries, or to the salivary glands, or to both, as shown here. Or they may just remain in the midgut. If they may remain in the midgut, they won't be transmitted to the next host. If they remain in this, if they move to the salivary glands, they will be moved out into the next host the next time she feeds, because the salivary glands are the site of things that go from tick into the host, as you see here in this diagram. If they move into the ovaries, which some pathogens can do, then they'll be transmitted to the offspring of that tick. And so, without going into any more excruciating detail than I already have, we should know that different pathogens make these migrations at different rates and at different times during the feeding. And that some of them move from salivary glands, from midgut to salivary glands. Some move from midguts to ovaries. Some move to both, and some move to neither. And the ability to move into those different organs will determine whether that vector, that tick, can transmit the pathogens that it's going to pose to us. And that's the reason that some ticks transmit some pathogens and some ticks don't transmit those same pathogens. It's also the reason that we say it's fundamentally important to know how long a tick is fed to be able to assess its risk. So here's a diagram showing the feeding process of ticks going from unfed through feeding on day 7. They start out in a situation we say flat, they're unfed. They become partially fed and then eventually engorged. And the risk of transmission, that is the movement of pathogens from that tick to the host that it's feeding on, increases with time. And there's some critical time when it becomes more likely that the pathogens are going to be transmitted. And this is directly as a result, going to that last diagram, as a result of the spirochetes moving, the Lyme disease spirochetes moving out of that midgut into the salivary glands after the feeding has taken place on the previous host. For other pathogens, like Powassan virus, now shown here at the bottom, that process can take place much quicker. In some cases, the viruses can move very quickly out of the tick and perhaps and into the new host, perhaps in as little as 15 minutes. And so it's important to understand those transmission dynamics to understand risk. So what can we say? Well, we can be sure that every tick-borne disease starts with a tick bite. It requires the transmission of a pathogen associated with that particular tick species, and that not every tick individual is infected. So not every representative of any particular species is infected, and that the risk of human infection, that is the likelihood that a human will acquire the infection from that fed tick, increases with the feeding time. That's the conclusion of that very in-depth biological discussion. Now, as with tick biology, there are some excellent resources for understanding, for example, the symptomology and the treatment and different aspects of diseases themselves. And one place that's sort of safe to point people is to the Centers for Disease Control. Now, some listeners will perhaps not recognize the completeness of the information at the Centers for Disease Control, but nonetheless, it is a source that you can consider for information about the pathogens. Because my description here will be relatively brief, and I would encourage you to go on to either the CDC page or Massachusetts Department of Health or other authoritative sources for information about the pathogens. So, by and large, the biggest concern with the bite of deer ticks is transmission of the Lyme disease pathogen. Borrelia burgdorferi is the causative agent. It is the most common tick-borne disease. It is, in fact, the most common vector-borne disease in the United States. So all others, Zika virus, West Nile virus, all the mosquito pathogens, basically pale in comparison to the transmission rates of Borrelia burgdorferi by deer ticks in the northeastern United States. And I'll show you stats and where you can access the stats at the end of this talk. It can be transmitted by black-legged ticks as well as ticks from the West Coast, including possibly the western black-legged tick. Recently, we did a study where we found, we looked at human biting ticks that were sent to us for testing from the state of California, Oregon, and Washington. And we found that there was actually another tick, Ixodes spinipalpus, which may be involved with enzootic transmission or epizootic transmission of the pathogen in those states. There's seasonality to the transmission of Lyme, as there's seasonality to transmission of most diseases, tick-borne or otherwise. This is a diagram showing the submission of ticks to the Laboratory of Medical Zoology to Tick Report in the period of early spring 2014 through the winter of midwinter of 2015. And you can see that there's a peak of submission of adults in the spring. There's another peak of adults submissions in the fall. And then there's a single nymphal peak that occurs in the early summer. And I told you that before when I showed you the 2-year life cycle. That's the nymphal cycle that we see. You may note that there's very little evidence of larval ticks, and there's a couple of reasons for that. One is the ticks are so very, very small that very often they probably go undetected and or don't get sent to us. The other is there may actually be some fundamental behavioral differences of larvae in terms of where they're questing, and they may actually not feed on humans at the same rate that they feed on other mammalian hosts. But you can basically, by the same notion that you can think of the distribution of geographic distribution of ticks as representative of the risk of exposure to pathogens, so too you can think of this temporal distribution. The seasonality for exposure is when the ticks are abundant, then the risk is higher. And so in these months where it's low, or these months where it's low, probably due to snowfall in that year, the rates of transmission will be lower. Now, the other thing I would emphasize of this is that the canonical explanation is that most Lyme disease transmission or most Lyme disease detection actually takes place in here, not as a result of tick bites that occur in this period, but as a result of tick bites that occur in this period, attributable to nymphal transmission. And the thought is that even though the rate of infection, roughly 25 to 30% of nymphal ticks, is lower than the rate of infection in adult ticks, which is around 50%, that these ticks are the ones that predominantly transmit to humans. And the reason, the logic for that is, sorry, I had to skip a slide there to show this, is that the nymphal stages are much, much smaller, and they're much more likely to feed to that critical point when the pathogens move out of the midgut and into the salivary glands and into the host. And because they're so much smaller, they have a higher likelihood of doing that before they're detected. I would just point out that what we see from ticks that are submitted to Tick Report is that there are still a large number of ticks in the spring and the fall that are fully fed, that is, that have fed on people for more than the period that's thought to be sufficient for transmission of pathogens. And so we often wonder whether at least some of the cases that are reported here and attributed to nymphal infection may in fact have been due to adults that fed on them maybe just a few weeks earlier, and that we may be missing some transmission that takes place in the late fall because generally the thought is that transmission of Lyme disease takes place in this period, and we're not looking for Lyme disease. That is, in a physician's office, perhaps physicians aren't as acutely aware and not as likely to test or to reach the diagnosis. It's speculation at this point, but it's just another one of those cases where the entomological evidence doesn't necessarily match up with the public health evidence, and it warrants further explanation. This is just I skipped over this slide now twice, but this slide just shows that 2-year life cycle and emphasizes the idea that because the nymphs come prior to the larvae, so here it's not shown as a cycle, it's shown linearly, but because the nymphs come before the larvae, they're actually introducing the infection into the larvae that will carry over to those next set of nymphs that then transmit it to the larvae. So it emphasizes the importance of the nymphs coming chronologically in an annual cycle prior to the larvae. So I mentioned before that not only Lyme disease is not the only concern. There are also relapsing fever-like Borrelia. There's a Borrelia pathogen called Borrelia miyamotoi. It's a relative newcomer in the northeastern United States. It was first identified in Asia. It now seems to have a limited distribution here. In fact, some recent analysis that we completed suggests that the infection rate or the risk rate in coastal areas of Massachusetts is probably 2 to 3 times higher than it is for sites inland, because the infection rates are higher in those ticks. We don't know precisely why that is, whether it's has to do with what the ticks are feeding on in those areas or some other factor, perhaps climate factors still need to be sussed out. So we say that this one is present but not common throughout the range of Ixodes. And by that we mean that if 50% of the adult ticks have the Lyme disease spirochete, maybe 1 to 2% of the adult ticks have this spirochete. There are also pathogens associated with diseases called ehrlichiosis and anaplasmosis, and we group these together because there is some confusion. Anaplasmosis was previously referred to as ehrlichiosis as well. They've since been distinguished. So there are actually at least three associated bacterial diseases associated with, respectively, Ehrlichia chaffiensis and Ehrlichia ewingi. Ehrlichia chaffiensis is transmitted by Lone Star ticks, and Anaplasma phagocytophilum was previously known as Ehrlichia phagocytophilum. Its name has been changed, and it's in fact a separate parasite and infects different cells within the mammalian host. And more importantly, or perhaps most importantly in the context of the present talk, that is the only one that's transmitted by deer ticks. Ehrlichia chaffiensis is not transmitted by deer ticks. Going through the list of other pathogens associated with deer ticks, babesiosis is a disease that's caused by a malaria-like pathogen. Malaria-like because it's not a bacteria, it's a protozoan, so it's more akin to an amoeba. It's still a single cell, but it has a life cycle that's much more similar to malaria parasites than to a bacterial parasite. It's transmitted by black-legged ticks or deer ticks, just like Lyme disease. It has been associated with blood being found in blood banks, and so comes through transmitted occasionally that way. It's not as common as Lyme disease, but it could be as high as 10% in certain endemic areas. So it can be a concern. And it's also a concern because since it's not a bacteria, it's not treatable by the same antibiotics that will treat ostensibly all of the other bacterial pathogens associated with deer tick transmission. Powassan virus is the most common viral pathogen, but still relatively rare, much less than 1% in what we've seen of the ticks that we've tested. There are two different types. Type 1 is the canonical Powassan virus. The disease was initially named for the town in Powassan, Ontario, from where it was originally identified. It's actually been found throughout the continental United States, associated with several different tick species. But in recent years, it's been most associated with something called Ixodes cookii, not a deer tick, but a cousin of the deer tick called the woodchuck tick. Because it's been mostly in woodchuck ticks, it hasn't been a primary concern. Woodchuck ticks feed on woodchucks in woodchuck burrows. And since most of us spend very little time in woodchuck burrows, it doesn't pose a serious risk to us. In recent decades, we found a second type, type 2, which is also called deer tick virus. And as the name implies, it is transmitted by the deer tick or the black-legged tick. It seemingly has the same clinical manifestations, can cause the same encephalitis that the type 1 Powassan virus can cause. It's found mostly in the northern U.S., correspondent with the distribution of deer ticks. And again, these are not published results, but I can say anecdotally that we have seen a number of cases of people having been bitten by ticks that are positive for Powassan virus, and yet they show no signs of infection or illness. And so whether this pathogen is pathogenic to a small subset of the people it infects, such as West Nile virus, or not, is something that remains an open question that our group and others will be interested in research. So I have, so I want to finish just by inviting you all to attend the next webinar to be offered, which is on April 10th. Here's the contact information. This will be on the slide. It's also posted on the webpage. But before wrapping up entirely, I wanted to do something because I think it would be of interest, which is to point you to our webpage, which is tickreport.com, and then show you what you can do if you were to go and visit that webpage and click on our statistics page. So one of the questions that people will have, or what are the incidence of these different pathogens? How often are ticks infected with them? And I'd like to point out to you that on our passive surveillance web page, Tick Report, you can view the statistics of ticks that have been sent to us by clicking on our stats page. There'd be a small disclaimer where you agree that you're not going to use the information for nefarious purposes. And then you'll be taken to this page, which shows the summary of the 48,171 human-biting ticks that have been sent to us. And you can see the breakdown of the different pathogens that have been found in those ticks. I'm sorry, I have to move a window so that I can see what I'm doing. There are 40,000 ticks that have been tested, and among those 10,000 ticks, 10,696, or 26%, were positive for Borrelia burgdorferi. And you can go and you can see these statistics for all these different pathogens, including some of the very, very rare ones, like Powassan virus, which I told you before is much less than 1% of the ticks that we've tested. And because the data are, most of our ticks still come from Massachusetts, although we have ticks from all 50 states. If you were to restrict this to just Massachusetts, and for purposes of our conversation today, just deer ticks, and then search again, that would give you the numbers for the pathogens that we talked about today, as well as some additional ones. So, for example, I had told you that Borrelia miyamotoi, the third line here, we've tested now 11,000 ticks, 231 were positive for an infection rate of about 2%. Likewise, Babesia microti, statewide, about close to 7% infection rate. Anaplasma, the agent of anaplasmosis, around 6%. And then you can see the numbers for other things. We've actually seen a slightly higher than the national average, where, as you recall, that number was about 0.19. It's about 0.43% for Powassan virus here in the state. Now 12 positive ticks out of 2,778. I want to emphasize that there are some caveats about looking at these data. For one thing, these are mixing the results for both nymphal ticks and adult ticks, which is one of the reasons these are a little bit lower. So if this were purely adult ticks, the number would be slightly higher. If it were purely nymphal ticks, it would be something slightly lower. But because they're mixed in this survey or in this query tool, it sort of takes the mean of the two. Nonetheless, this gives you an average annual risk for the different pathogens and answers a number of questions. Now below is a case by case. These are all blinded cases, so you can see the results of individual ticks. For example, this tick from Scituate. You can click and actually see these anonymous stories of a tick that was sent to us from a 9-year-old female from Scituate, Massachusetts, which was positive for Borrelia burgdorferi. And there's a picture of the tick, the condition of the tick, and the feeding status, etc. But I just wanted to finish by just saying that if you have questions about the incidence of pathogens, we have a shared public database where you can get that information directly. And maybe, since I've now opened this can of worms, I will speak to this one issue at the top. So you may notice that there are several different Borrelia species that are associated with ticks in general. Borrelia burgdorferi, Borrelia miyamotoi, Borrelia mayoni. And we have reason to believe, because of the global picture of Lyme disease, that there may be other species of Borrelia that have yet been identified, or maybe are causing disease and they don't fit canonical clinical definitions or test positive for existing tests. So one of the things we do with every tick we test is, in addition to testing very specifically for the known grouping of bacteria associated with Lyme disease, we also do a general species test that will theoretically identify any Borrelia species in the universe, known or unknown, for found in ticks. And by doing so, we can detect things that have yet to be identified, and then in subsequent work we can characterize those further. And so what you're seeing here is that when you see the 42% for the Borrelia general species, that's the summation of all the specific examples of the specific species. And some people sometimes get confused by that. It's basically there for us biologists to get a little geeky about. And I can tell you that in the 44,000, whatever the number was, ticks that we've looked at, we have found some examples of some what we believe are some rare Borrelia that we want to go back and further characterize. So the work is ongoing, and the surveillance, the passive surveillance, is an important tool, and that's why we emphasize it here. So with that, I'm going to go back to the summary slide, and thank you for your attendance, and apologies again for going long due to the technical difficulties. And I'll ask if Jeffrey